Good afternoon. My name is DuBois Bowman, and I'm privileged to serve as Dean of the University of Michigan School of Public Health. Thank you for joining us for today's discussion on COVID-19 vaccines, especially at a time when our nation is facing unprecedented challenges. Some of you may have joined our COVID-19 vaccine webinar in early December. Even in the month since that time, there have been remarkable developments. 2 vaccines have been approved here in the United States, and millions of doses have been dispersed and administered. It's a remarkable scientific feat, and my colleague, Dr. Arnold Monto, has played a key role in this. For me, watching the first round of COVID-19 vaccines be administered to healthcare workers brought a sense of optimism and hope, and knowing that we still had challenges that lie before us, beginning to see the light at the end of the tunnel, so to speak. But the vaccine rollout has also brought with it many questions. How do we know the vaccines are safe, and for whom? How long will immunity last? Do the approved vaccines offer the same level of protection against some of the new strains that are emerging that we've been hearing about in recent weeks? And to help answer some of these questions, we're joined today by University of Michigan School of Public Health professors and infectious disease experts, Dr. Arnold Monto and Dr. Emily Martin. Dr. Monto is a world-renowned epidemiologist. He serves as acting chair of the FDA's Vaccines and Related Biological Products Advisory Committee, or VRBPAC, which is advising the FDA on the authorization and licensure of COVID-19 vaccines. Throughout his career, Dr. Monto has been involved in pandemic planning and emergency response to influenza and other respiratory virus outbreaks, including the 1968 Hong Kong influenza pandemic, avian influenza, SARS, MERS, and of course the coronavirus pandemic. Dr. Martin leads CDC and NIH-funded studies of influenza vaccines in ambulatory care, hospital, and also community settings. And during the pandemic, she's been using COVID-19 public health data to help inform mitigation and policy efforts at state and local levels. I'm grateful to have the expertise of researchers like Dr. Monto and Dr. Martin here within our School of Public Health community, especially during this pandemic. And I'd like to thank both of you for taking time to share your expertise. And with that, I'd like to turn it over to Dr. Monto.
Thank you, Du Bois. I'm going to talk for a short time about some of the developments since we last had a webinar a little more than a month ago, because as Du Bois said, we've had 2 vaccines approved. The vaccines have been rolled out, and there have been a number of developments in the overall epidemiology globally of this virus, especially concern about new variants. I'll try to do several things more or less at the same time to go over the story and to use what we heard at the advisory meetings to give you a little background about some of the debates that have arisen globally about, for example, the schedule of vaccination. Should we delay the second vaccine? And also something about the dosage issues. Can we spare doses and use less vaccine for more people? And try to summarize in terms of where we are now. I'll also try to talk about the future in terms of new vaccines, which are being evaluated right now. So let's get going and look into some of these developments. Let's see. I don't seem to be able to move my screen. Let's see what's, there we go. Okay, Now we'll give a little bit of a review since last time. As I mentioned, the reason we've been so successful at developing the vaccines so rapidly is that one component of the virus, the spike protein, seems to be related to development of protective antibodies. So the target of all of vaccine production has been the spike protein, development of antibodies to the spike protein, which have fortunately been pretty well correlated with protection. We don't know that for sure. Once we know that we have a real correlative protection, it might be possible to be able to start approving vaccines just on the basis of their producing antibodies to the spike protein, but we're not there yet. So here are the three kinds of approaches that are being used. The one that's at the bottom, the mRNA path, is the one that is being used now in the 2 approved vaccines. The middle one is using an adenoviral vector to get the RNA that encodes for the spike protein delivered to the body, which again produces antibody. And the third approach, which is number 1 listed here, is the one that is coming along most slowly, and that is a more traditional approach to developing the spike protein in a cell culture system and delivering that protein to the body for antibodies to be produced. And here are, again, as a review, the Moderna and the Pfizer BioNTech vaccine, the mRNA vaccines, which we now already have approved. The next ones that are going to come up, Janssen is probably going to be coming up next. And then the AstraZeneca vaccine, which has already been approved in the UK, which are the vectored vaccines. That's how the mRNA gets into the body through delivery through a vector. And then the protein vaccines, which are going to turn out to be adjuvanted in addition. Now, this is the agenda, the first page of the agenda for the December 10th vaccine in which the Pfizer product was approved. And for those of you who watched the procedures at the meeting, you need to realize that the agenda and the questions that are asked are determined by FDA. The advisors are more or less a jury, who are brought in to just discuss and vote on recommending approval. And this is the first page of the agenda. And you can see that what FDA was doing was setting the scene for an emergency use authorization to establish the fact that there was a need for this authorization, showing what was being done on epidemiology, that the pandemic was severe and required emergency use. Now, from the data that were presented to us, which you can find, this is all publicly available, one of the more interesting features was the fact that from day 0, which is when the first dose of the Pfizer vaccine was given, there wasn't that much difference between the red, which are infections in those that received the vaccine, and the blue, those in those that did not receive the vaccine, and the blue, which are those who did receive the vaccine. But by day 14, there was already a separation. This is before the second dose was given. Second dose for this vaccine is on day 21. And then if you look at the data here, you will see that at the bottom, there was after 7 days after the second dose, and this is before the antibodies should have been kicked in for the second dose, there was already 90% effectiveness, vaccine efficacy of the vaccine. And that's one of the reasons why there has been a suggestion to delay the second dose, because there is significant efficacy before that dose really kicks in. And but that was never really tested. And the numbers are relatively small compared to the overall numbers, which is why FDA has said that we're not ready for this kind of an approach, which may be used in other countries. And then we had the vote. And the vote here to this question was 17 yes, and no was 4. And 2 people said they didn't think we were ready yet because they wanted the time from the last dose to be a bit longer, a couple of months. But most of the reason that there was a negative vote or abstention was because of the issue of vaccinating 16 and 17-year-olds, because there wasn't as much data. One of the pediatricians voted in favor and the others voted no. And in private, they mentioned that if the 16 and 17-year-olds have been removed from the question, then they would have voted yes. So there's no reason to take the no votes here as being any disapproval of the process. And then we had a week later, and I just hope going forward, we don't have approvals that close to one another. It takes a while to recover from a nine-hour stint working on dealing with a large number of people and getting things going in an orderly fashion. And here we considered another issue in addition. And that issue was what happens to those who are in the placebo group. And this is the Moderna vaccine, who are able to get vaccine because their priority group has come up. This is an unusual situation, only is in place when we have an emergency use, because ordinarily vaccine rollout is not so quick, and we have no problem in retaining people in the placebo group, because the placebo group is very valuable for comparisons in terms of side effects with the vaccinated group. So there was a considerable discussion about this. The thing that was most impressive about the Moderna vaccine was they had a number of more severe events occurring in the placebo group than with the Pfizer vaccine. This was mainly because of timing and where the testing was done. And here they had 100% prevention of severe disease. Again, the numbers are small, but certainly this is impressive. And they made the point that it was hard to ethically say that the placebo group shouldn't receive vaccines when they knew they had such high efficacy. The efficacy against just laboratory confirmed symptomatic disease was in the same neighborhood, in excess of 90% for the Moderna vaccine and for the Pfizer vaccine. The separation, you can see it took a little bit longer to see the separation appear from the initial dose. And here the second dose was given at 28 days. Why 28 days versus 21 days? There's really no immunologic reason for this difference. This was just based on what was established in the protocol. And one of the problems we have with the rapid rollout of these vaccines is the fact we spent less time in trying to figure out exactly what the appropriate dose would be and the timing. If we weren't under the gun in terms of rolling out vaccines in the face of a severe pandemic, this would have taken a lot more time. And the positive is we've got the vaccines. The negative is we probably may want to tinker a little bit with the dose, but you can't beat 90% effectiveness. So the consensus right now from FDA is we go with what we've proven, and we continue to use what we have approved. The other issue is the half dose. And again, they chose 100 micrograms for this vaccine because 250 micrograms were a bit more reactogenic, and we'll talk about reactogenicity in terms of sore arms, especially afterwards. And they picked 100 because it was, if you look at the second, the GMTs for 43 days post-vaccination, that's 14 days after the second dose, and you look at 50 micrograms, it was 73,000, 734,000 versus 811. So you got more tighter for the 100 microgram dose, which is what has been chosen to be put in the vaccine, than with the 50, but it isn't that much more. And that's one of the reasons why it was suggested maybe we could use half a dose. You can see a big drop if we go down further, but look at the numbers that were involved. These were 15 individuals on which these numbers, each of these distributions occurred. So it's pretty hard to know what was better and what was not better. And probably what should be happening, and there's a talk about doing this, is doing small studies to evaluate whether we can fiddle with the dose, but we go with what we've got and approved in the phase 3 studies right now. We also had this other issue, which we were asked to discuss, and that is the unblinding issue. And what would happen with the placebo groups. It's a technical issue, but it does affect analysis going forward. And you may be hearing more about this as months proceed in terms of licensure, but because one of the requirements for licensure is 6 months of observation in the placebo-controlled trial design. So we're going to be hearing more about this going forward. And then the vote, same question, and we had nobody voting against this time. And I made the point closing the meeting that we shouldn't take a difference in vote to indicate preference for one vaccine or another. It's just that a time had passed. We didn't have the pediatric question come up in particular, and we all felt enthusiastic about the efficacy of these vaccines. So what's coming in the future? Janssen is probably, according to what we read in the press, going to come up in February, which is a 1-dose delayed second dose vaccine. So there won't be an issue of when to give the second dose because it's been delayed. It's going to be delayed in their design by several months. And the AstraZeneca vaccine, which has already been approved in the UK, and then trailing behind the Novavax vaccine, which is going to be a little more traditional in terms of what is delivered to the body. And here again is just a schematic of the vectored vaccines and the protein-based vaccines, which are coming along. This is all being run through, the Pfizer vaccine was not being run through the NIH Data Safety Monitoring Board. The Data Safety Monitoring Board is involved in the vaccines that are currently still in development. So they are watching this, and they are the ones that were responsible for the clinical hold several months ago. So things have happened so rapidly, we almost forget that the AstraZeneca vaccine was on clinical hold for several weeks because of one side effect that needed further investigation. And we expect the Novavax vaccine to be producing about the same level of antibodies. It's going forward. And they only now started the phase 3 clinical trial. So it'll be several months before that is ready, ready for submission for approval. Safety assessments. We've been concerned about safety since the start, a major concern with the new vaccine and the side effects that we know about. And there have been most side effects to vaccines occur within the first six weeks. For example, the Guillain-Barré associated with the influenza vaccine. And basically, we have seen very little, but given the known side effects associated with administration of the vaccine, sore arms, much more likely to occur than with our flu vaccine, at least our current flu vaccine, which is highly purified, much more similar to the herpes zoster shingles vaccine, where you do get sore arms and you do get side effects in terms of low-grade feverishness and other myalgias and the rest in a small proportion of individuals, very infrequent. Now, what came up right before licensure or approval of the Pfizer vaccine was this issue of anaphylaxis. Now, we know that anaphylaxis does occur very rarely with any vaccine, and that's why you need to stay at the site of vaccination for several minutes, 15, 20 minutes before. CDC just came up with a summary of 11 cases per million of the Pfizer vaccine. Moderna vaccine seems to be less, but we're not sure because it started out later. So, but one case at least has been reported with the Moderna vaccine. Now, I showed this one before. I just wanted to say that the rollout, how the vaccine is utilized, is not a function of FDA, but more in terms of CDC. We may want to talk about that, but this is a very complicated situation. We're only realizing that with our healthcare system in the United States, it's not ideal in many respects. And it's certainly not ideal in terms of the rollout of a vaccine, which needs to be given to the entire population. There are other models, the UK model, where everybody is part of the healthcare system and they can recognize those with underlying conditions easily. The situation in Israel, which is the most vaccinated country probably in the world right now, where everybody has to belong to an HMO and they've got all their data. Those are the ideal situations. We don't have the ideal situation in the United States, but we work with what we've got. And as more vaccines come online, we will be able to get them into people's arms and be able to answer a lot more of the issues about duration of antibody and things like that. We now will really look into some of these other issues, and I'd like to turn over to my colleague, Emily Martin, who will lead us through these questions. And thank you for listening to me ramble on about all these issues. I hope I've been able to help in trying to deal with these rapidly moving events.
Excellent. Well, yes, let's go ahead and kick off the Q&A portion. And Arnold, if you can, there you go. Perfect. So
I'm slow, but
that way
I
we can
figure
see
it
you.
out.
Thank you for those who submitted questions in advance in the registration. We saw a few common themes in the questions. We're going to start with addressing some of those first. But in the meantime, at the bottom of your screen, there's a Q&A button on Zoom where you can submit additional questions. There are people that are going to be watching for those questions, and we'll get to as many of those as we can as time permits as well. So to kick it off, I'm going to start with a hot topic. So you mentioned the debate on the 16 and 17-year-olds that came up during the VRPAC meeting. From my mind, the 16 and 17-year-olds, they're not a high risk of severe disease population, but they're an important population if you think about transmission. They're very mobile. They interact a lot. They have jobs where they may be interacting with the general public. What is your view on vaccine safety with the two products that are on the table right now and using them in 16 and 17-year-olds?
Well, the interesting thing about the 16 and 17-year-olds is that we had two things going on at the meeting. One was the standard need, especially among pediatricians, to demonstrate that vaccines which are given for adults work the same in children. The other thing was just not paying attention to some of the data that was presented to us, because Pfizer had already done some studies in 12 to 15-year-olds where the vaccine had performed just as well as in adults. So I think we will see bridging studies which will allow the vaccine to be given to younger individuals. And the question will come up about prioritization. I recall that in the past flu pandemics, we've had discussions about who should be vaccinated first. With flu, as you know, Emily, school-age children are the vectors in the population. They spread it in the population. Should we prioritize them? Because then we can really bring an epidemic under control more rapidly. That's a discussion we haven't had here because we haven't seen that much symptomatic infection in young individuals. But boy, the asymptomatic infection is playing a much bigger role than we ever thought with this virus, this weird virus.
Right. And actually that brings up it kind of alludes to another question that we've gotten a lot of comments about, is learning more about how the ACIP process worked, the committee at the CDC that decides how vaccine prioritization, like how did we get to where we are with prioritization? Because there are a couple different ways to approach it, right? You can, as you mentioned with the, efforts to vaccinate school-age children against influenza, you can approach it by vaccinating the people spreading the disease, but may not be that impacted themselves as severely. You can vaccinate your people that are most likely to get severely ill first, or you can, as they did to some degree in ACIP, focus on your critical infrastructure. So vaccinating the people that you need to keep your hospital. You don't want mass amounts of your healthcare workers calling in sick in the middle of a pandemic, right?
Or the nursing home issue, which one of our colleagues got very much involved with discussing.
The nursing home, yeah, and so it's kind of a mix of both. I kind of listed out three options, and it's a mix of number 2 and 3. It's you're getting your infrastructure, and then in your frontline workers, and then you're getting nursing home residents. You have some age-based strata to try to do it by risk as well, and so it's sort of a hybrid of two different strategies, which is complex operationally, but it does kind of meet two needs at once, which there are benefits to doing that as well.
Well, I think now we've really had a demonstration of the different roles, and it's unique to the United States, of the FDA, which approves. Basically, it says who can get the vaccine, and doesn't go into any detail. FDA has approved vaccines that have been rarely used, but it's ACIP that really says how the vaccine should be used and makes recommendations based on evidence, and then goes into details. And the question in my mind is whether there is not too much detail in terms of who should get the vaccine here, in terms of our healthcare system and their ability to deal with the rollout. Because I'm concerned about, in 1C, people who have underlying conditions. I think I've alluded to that. How do people recognize who has underlying conditions, check on who has an underlying condition? With the flu recommendations, they used to have a recommendation for people with underlying conditions. They found it was unworkable, and they basically went by age. So you've got all of those logistic problems in addition to trying to get things moving. And our healthcare system is fragmented, as we know, and under our state health departments, and especially our local health departments, are under-resourced to do what it needs to be done here, which means we really have to fall back on what we have.
Yeah, and I wanted to mention to that end, there's a lot of people that have asked related questions about how to find out where we are in the vaccine distribution process, how to get instructions on where to get a vaccine, who to call. And you know, the way the U.S. healthcare system works, it either lives with your primary care provider or it lives with your county health department. Now, the healthcare systems, the primary care providers, are still getting up to speed on how to mobilize at that level. And so for the best source of information to go to is going to be the website for your county health department to figure out where they are, various health departments are in the process and how it's going. And then, you know, it's going to be the work of kind of announcing as they move through the phases, the priorities. I think Michigan's decisions to start changing their prioritization structure to expand things is going to move things faster here too.
And we have a lot of people who now are authorized to get the vaccine going down to age 65 rather than age 75.
Yeah, and then one of the tricky things will be to balance the needs of communities that are more integrated into communities that have got big health systems or sort of big infrastructures, and then the needs of communities that are further out that have more distribution challenges and infrastructure challenges, for sure. I'm gonna take us back to safety, because I feel like there's a lot more safety questions that we started out talking about safety, and then I veered into operations a little bit more because I like
well, that's what we live with. So
yeah.
So really safety, aside from the anaphylaxis, safety has really not come up. We've heard much fewer reports about side effects, which
for sure,
and really concerned about.
And the allergic side effects that have been that have been discussed are right now it's 0.001% of the time.
Yep. 11 per million.
Yeah, and so interesting to see that. So a lot of this, I think, discussion around safety comes from the fact that there's so much talk about how fast the whole process happened, because in warp speed makes it sound like you're just like hurtling through space developing vaccines at the same time. But
those of us who watched, who were Star Trek aficionados recognize the term immediately.
Exactly. And so, but you know, the terminology and the way of it kind of leaves behind, in my mind, the fact that these are based on technologies that have been in development for, and honestly, the flu vaccine field has kind of propped up some of these things, but these are vaccinology strategies that have been around for a while and have been in development for a while. And so it's, you know, what do you think about speed, whether there's been a kind of a speed versus safety trade-off?
Well, I don't think there's safety concerns that we really have to be worried about when we know we have a 95% vaccine efficacy. there is always a possibility of infrequent side effects. We typically don't figure them out until we have the rollout of the vaccine and the vaccine being used in millions and millions of people. As long as those side effects are not severe, this is not really a concern because we need to deal with risk benefit. And if there were much of a risk, we know it as the vaccine gets rolled out. We usually don't see those because of numbers in the clinical trials, but only after the fact. The one thing that there was a slight signal about was Bell's palsy in both of the trials, maybe one more in the placebo group, in the vaccinated group than the placebo group. The problem there is this is something which occurs naturally in the population in the unvaccinated. So there may be a slight excess, but again, it's so rare that you need to get millions of people vaccinated in order to be able to show there is a slight excess, which means that really the benefit so far outweighs the risk, that in the face of what we've got going on, people should get vaccinated without concern.
Yeah, it also, I'm impressed by the fact that I think modern technology and mobile technology and the fact that everybody can do there's the V-Safe program now, where all of the, everybody who gets vaccines is doing regular reporting on their side effects and any effect they've had from the vaccine for weeks after they get it. And we're getting that data in real time now. Instead of a passive system for every other vaccine we've rolled out, we wait to find out if we're hearing reports from doctors or hospitals about there being a problem. I think doing it at this granular level with this amount of data collection, we're going to find out about, if there are any safety issues, we're going to see them really, really fast compared to where we would have been years ago if we had done a similar kind of mass campaign.
But I alluded to some of the issues about the fast rollout, and that is they had to choose the right interval between the two doses and how much to put in the vaccine quickly. This usually would take quite some time with dose finding and looking at different intervals. They just picked intervals, and we're really locked in, and appropriately so, with the dose and the interval that we've got right now, because that's all we've got data on. In parallel, there probably will be studies to see whether we can not optimize, because I don't think we can go higher than 95%. But to spare doses and allow more people to be vaccinated, but that has to be done in parallel and in small groups where you have an efficacy endpoint, because we really don't have a correlate of protection yet. We can't just use antibody as a surrogate for protection. And then we have the duration issue, and that's gonna take a while to figure out.
Yeah, and it's interesting that a lot of the dosing schedules for other vaccines have evolved over time as we've seen them be used and see what works in the population.
Exactly.
And we're not used to tackling that question. We're used to tackling that question after a vaccine's been out for 2 or 3 years now.
Exactly.
You're changing the dose or changing the schedule. All right, last safety question. Before we tackle some other, I want to get into the variant strain questions. But so let's talk more about pregnant women. So there has not been a specific study of pregnant women, but pregnant women were included.
Never is a specific study of pregnant women, because especially the pharmaceutical companies don't want to tackle it because of liability issues and all sorts of things.
Yeah, but
it's always trying to figure out, except when we get into a recommendation for pregnant women, as with influenza. For many years, we didn't use influenza vaccines hardly at all in pregnant women until well, really the 2009 pandemic, where we saw that pregnant women were at particular risk for if they got influenza. And now we recommend vaccinating pregnant women. We know it's perfectly safe.
Yeah, and I think similar, looks similar for the COVID-19 vaccine as well. With pregnant women, pregnant people react similarly to the rest of the population that has been looked at, and no long-term effects are expected with that, just like the rest of, expected to work just like the rest of vaccines.
And CDC's got permissive recommendations.
Yeah.
In other words, if you're in a risk group and you're pregnant, you can get the vaccine.
So, all right, switching topics, let's talk a little bit about the new variants of the virus. So there's the B.1.1.7 variant in the UK. There's a similarly structured variant reported in South Africa. And got a lot of questions about that. I can tackle that one, or you can take a crack at it.
You handle that one, because you're much more molecularly inclined than I am.
Yeah.
My initial reaction to the UK one was, oh, we've had variants come up from the start, and it came up just when they were having some political problems with shutdowns in the UK, and I wondered whether the news was overblown, but it seems not, that this is really different.
It does seem to be different. Well, and one thing I want to make sure everybody understands is that viruses, especially these kinds of viruses, are always mutating and changing. And so it's not like something is, this is sort of a natural process. This is kind of evolution, a natural evolution at play in these viruses. The changes for these particular variants, they seem to be operating in a way where they do seem to make it more transmissible. You can see it in the epidemiology data, and you can see it in the molecular data now. It makes it so that the virus is particularly good at sticking to the cells that it needs to replicate and produce and go on to replicate and infect other people. And so it probably makes it spread faster because it makes infection more efficient. But it doesn't change the way that, at least so far, these two, doesn't change the proteins of the virus in a way that makes our body react differently to them. To our body, they look similar to the vaccine, the proteins that come out of the vaccine process. They look similar to other versions of SARS-CoV-2. And so at this point, there's not any concern on the table about the vaccine not working or various treatments not working. It also doesn't look like they're more severe, they're causing deaths at a higher rate. I think they are just spreading faster. So I think it's going to mean we've got to be more careful with our public health mitigation. But the vaccine becomes even more important because the vaccine is still going to work just as well against the variants.
And if there is any issue in terms of vaccine efficacy, we'll probably hear about it first from the UK. They're rolling out the AstraZeneca vaccine along with the Pfizer vaccine. And they've got really domination of the new variant, which actually emerged there in September. It's not really anything that just recently came up.
Yeah, it really seems to have taken hold right as they were coming out of their restrictions period.
Right.
Difficult timing.
And basically, this is not something which emerged because of antibody pressure. That it's something that probably was a random event, and because it had an advantage, it took over.
Yeah, and I don't think, a lot of people don't realize we already have a huge kind of plan and infrastructure, an existing way to look at this, because we're so used to watching this for flu. We're used to watching changes for flu throughout the year and looking to see if it changes who's getting infected depending on who's getting vaccinated. And so we're kind of, there's a system already set up to watch this going forward, and it'll tell us. Right? I'm sorry?
Especially in Michigan, right?
Yeah, especially in Michigan. And so
6% of all the sequences in the United States are from Michigan.
For Michigan. And so, we're used to watching it and figuring out what it means for the vaccine. But then that also tells us, we're also used to doing, the US does such a good job of all the monitoring of vaccines that are already around. So how long does protection last? Are we going to need booster doses after a year or something like that? Or is the virus changing enough that we're going to need to update it? We can use the same methods that we use for the flu vaccine to figure that out. What do you, based on the what you saw in the FDA meetings, what do you think about waning and how long immunity is going to last from shot to shot?
As you know, we, in our household study, we've got data on 10 years since 2010, data on the seasonal coronaviruses. These are 4 viruses: common coronavirus, seasonal coronaviruses, 4 viruses that cause, in most people, common colds, occasionally more severe disease. And we know there are reinfections. We even have, as you are well aware, Emily, one individual who had 3 infections with that virus and was one of the first people in the group of households that got SARS-CoV-2. This would suggest that antibody is not going to be long-term, that we are going to need booster inoculations, but also what we know from other viruses such as flu is, and RSV in particular, that a reinfection, once you've got antibodies, is not as severe. So there's, again, no downside to be vaccinated. As a matter of fact, there's an upside, because if there is waning of immunity, you're likely to have a more mild disease if you do get infected.
So let's talk a little bit about, so we talked about a lot about effectiveness in preventing disease in the person that's vaccinated. Let's talk about effectiveness against transmission, which in the field of infectious diseases has been like the tricky question for a long time is, if the vaccine is preventing disease in the person, is it also preventing them from transmitting? And just to kind of clarify where this question comes from people before I bump into Arnold is you might be able to have an infection that's very mild and asymptomatic that you're able to spread, and that wouldn't necessarily show up in the initial trials of the vaccines because those were measuring to see who got sick. And so you could end up having being vaccinated by getting an asymptomatic infection that could still be transmitted. And I think data is still coming to show that. How likely, I mean, with 95%, I'm having trouble believing that, but they're still waiting to kind of see.
First, give us your opinion, because
I'm on the fence. So this is an incredibly effective vaccine. It is way more effective than I expected to see coming out of the trials. And so I'm very hopeful that it can prevent transmission. At the same time, we've already seen how well this virus works asymptomatically.
Yeah.
And so I think I do want to wait for the data to see whether transmission is possible, post-vaccination.
We've had to change our thinking totally about asymptomatic transmission since this virus emerged. There was little evidence with the original SARS virus that there was asymptomatic transmission. This virus is much more efficient at, less efficient in causing severe disease. So there are more asymptomatic cases, and those cases seem to transmit, but not regularly. And we still don't know why some people transmit more than others. And I think we're not going to be able to figure this out until we get more data. And again, it's certainly not a reason to get the vaccine. It's a reason to get the vaccine so we can figure it out better once we've got a more vaccinated population, because we'll never be able to say how to prevent transmission. But even if there is asymptomatic transmission, and you have a vaccinated person who gets infected asymptomatically, you're preventing symptoms. So it certainly should not be a reason not to get vaccinated.
Yeah, but that's also why people can expect to still see things like mask recommendations, social distancing, all of the non-pharmaceutical tools that we've been using to date are still going to be in place until a critical number of people start getting vaccinated and until we know more about how transmission works. So I'm going to hot topics again. So now that there's 2 vaccines, are there, well, I think talk about the different types of products. So Pfizer versus Moderna, there's 2 different vaccines, it's very similar technology. Do you think that there is a difference between the 2?
Well, there is clearly a difference between the 2 in the way they've stabilized the RNA. In terms of how you have to
meaningful difference. Somebody should go try to get one versus the other.
Well, I don't know that there's any real difference, except maybe because of packaging of the RNA, and there are proprietary elements here in terms of the anaphylaxis. I don't think we know, because Moderna followed the Pfizer, not quite as much has been used, whether we're going to see the anaphylaxis with the Moderna product as commonly as we do. It's not common, but with 11 per million, we're using so much vaccine, we'll probably have not statistically significant differences, but a bit of a trend there. That and the way the vaccine has to be handled, I think, are the only differences that I can see so far. Duration, we'll know that long term. There may be some tighter differences. All of these vaccines are being standardized in one NIH lab that we know well. So we will be able to see things long term. What I think is going to be very interesting is what happens with the other products. What we've heard from AstraZeneca is a bit confusing, because we've heard estimates of efficacy anywhere from 60-odd% with the standard dose to 90-odd% with half dose, which is not what's in their protocol. And that's one of the reasons why the AstraZeneca vaccine, which was supposed to come up for review in February, is probably going to be delayed to April, because things are a bit confused in terms of some of the results of the trials. I think the Janssen or J&J vaccine is going to be very interesting with the 1 dose, because and they do have in their protocol, that's in clinicaltrials.gov, so it's publicly available, that a 2nd dose will be given 3 or 4 months later. But there's an interval there, and we'll be able to see what happens, and it's almost like a booster dose.
Yeah, and so, you know, I think the U.S. is notable from the fact that we are solidly on these 2 mRNA vaccines for now, and we're starting to see a broader range of other vaccine technologies being used in other countries. But the data on these, I mean, these 2 vaccines are both coming out with these efficacy numbers that are definitely in the higher echelons that we see with vaccines.
Well, it's going to be interesting to see how the traditional vaccines fare, the Novavax. And one of the problems they're going to have in the US is to run the trials, because by the time they get going, I alluded to the placebo group and people in the placebo group wanting to get vaccine, wanting to be unblinded and get the vaccine. They're not going to be able to run the Novavax trial in the US for older individuals. And that was a requirement for the 2 vaccines that are currently under emergency use authorization. They're going to have to go outside the US, because would you, if you're over age 65, want to be in a placebo group when you can get one of the vaccines that's already licensed? So they're going to be going, and as you know, they've done RSV vaccines. in various countries in the world, they're going to go back to those countries in order to test in older individuals so they can maintain the placebo group. And we're going to have even more problems going forward. We're going to have, as we improve if we start changing dosage and things like that, it's going to have to be against an active comparative, vaccine against vaccine, because we're not going to be able to do a placebo trial in the US ethically.
Absolutely. And I think what's one of the things about kind of in the theme of safety and administration we haven't covered is vaccine hesitancy, and how in this age of social media and internet, how widespread vaccine hesitancy is, and how
we're polarized. We're a polarized country in so many ways. One of the things that we're polarized about is getting this vaccine, because there are some of us, including me, who are eager to get the vaccine. And there are a lot of people who really want to get the vaccine. And then there are some people who don't want to get the vaccine. And it's really a paradox, because those that want it bad.
Yeah, it's interesting. I mean, I do, having talked to some of my contacts that are more vaccine hesitant, I mean, the results from the trials about how striking the effect was and how fast it was, I think we're going to see it working. We're going to start seeing the vaccine working, and I think that's going to mean a lot to people once they start to see that it really does prevent deaths.
And because we're rolling out so much vaccine, they'll see that there is no safety signal.
Yeah, most definitely. So we talked a little bit about, one of the questions I often hear come up, and I've seen come up in the Q&A, was about people that have been infected previously. And I know you touched on reinfection, and this idea of why are we vaccinating everybody? Even if you've been infected before, and we know that that COVID-19 can cause long-term chronic disease, chronic issues that go for much longer than just the acute phase of the disease. I think that you've talked about the importance of reinfection. Kind of, it speaks to the importance to vaccinate everyone, not just those people who have not gotten infected already.
Yeah, and some of the people in the clinical trials were known because of the antibody survey they did at original vaccination were infected before, so there's no safety concern.
Yeah, so they've previously checked that. Yeah, excellent. Well, I see that we are at 2 o'clock. And so I want to thank everybody for joining us today. Unfortunately, I wasn't able to get all of the questions on the list, but we will continue hosting these.
You're so skillful at weaving them in.
I try. And my students have taught me well. So yes, so we're going to continue hosting the webinars regularly. Watch for more information from Michigan Public Health on vaccines, on all sorts of public health issues. And we'll be sending out a recording of the webinar over email to all registrants. And so feel free to share around, and we look forward to seeing you again.
Bye-bye.
Thank you.